Hello, everyone, and welcome to Advancing Biomarker Discovery for Disease Insights. I'm Iris Kolbatsky, Associate Science Editor at The Scientist, and I'll be moderating today's event. Before we begin the talks, I will share a few quick notes and tips to help you get the most out of today's program. As well as our fantastic lineup of speakers, I'd like to point out the interactive areas on the platform available to you. We'd like this event to be as engaging as possible, so please send your questions or comments at any time using the Q&A section to the right side of the live stream player. Our speakers will address these during the Q&A sessions following each presentation. We actively encourage discussion. However, please note that we do not allow solicitation of products or services and may remove any users who violate this rule. This event was made possible thanks to the generosity of our sponsors and partners. You can learn more about them and access their resources in the handout section as well. Now let's move on to our opening session. Andrea Di Pietro is a postdoctoral research fellow in the Neeson Laboratory at the Peter MacCallum Cancer Center, where his research focuses on tumor-resident T cells and gene regulatory networks driving immunotherapy resistance in melanoma. His work aims to enable earlier identification of non-responders and to inform the development of personalized immunotherapy strategies in melanoma. He holds a PhD in basic and advanced immunology from the San Rafael Scientific Institute, where he identified the antiviral role of TRIM22 in restricting human influenza virus infection. Following his PhD, he joined Monash University as a postdoctoral researcher investigating the epigenetic regulation of B-cell differentiation during acute and chronic infection. This work led to first author publications in Nature Immunology and Cell Reports. His discovery that chronic infection-driven epigenetic alterations impair antibody quality was recognized with the 2018 Careers in Immunology Fellowship from the American Association of Immunologists. A warm welcome to you, Andrea. Please take it away.
Hello everyone, and thank you for the opportunity to speak today. My name is Andrea, and I'm a senior postdoc in the Neeson Lab in the Cancer Immunology program at the Peter MacCallum Cancer Center in Melbourne. I have the privilege of working under the guidance of Professor Neeson and alongside with clinician researcher Dr. Louis Howe, whose photos you can see on the side of the slides. Today I will talk about melanoma and some of the results of our work on different cohorts of patients with either early or late-stage disease. And across these settings, I'll focus on tumor-resident T cells, their spatial organization within the tumor microenvironment, and how this might be harnessed as a biomarker for predicting melanoma relapse at early stage or resistant to immunotherapy response in a late-stage disease. Before I go through the results, I would like to begin with the acknowledgement of country. Peter Mac respectfully acknowledges the traditional owners of the land of the Kulin Nation. We pay our respect to elders, past and present, and we acknowledge the traditional owners of the lands on which our sites are located throughout Victoria. As many of you might know, Australia is one of the highest melanoma rates in the world, as you can see in the map. In Australia, melanoma is the third common cancer, and in 2025 there were an estimated 17,000 new cases of melanoma, which account for about 10% of all new cancer diagnoses. Around 1,500 people were estimated to have died from melanoma last year, representing roughly about 3% of all cancer deaths. So melanoma is still a major impact on our health system, despite being just one of the many cancer types in Australia. Clinically, melanoma diagnosis starts with dermatologists and other clinicians examining the skin, often using dermatoscopy and then performing a skin biopsy where is the suspicion lesion. So pathology then provides key features such as breast low thickness, ulceration, mitotic rate, and other features to then progressing into staging. For patients who present or progress to late-stage disease, diagnosis and staging extend beyond the primary lesion, and at this point we rely on cross-sectional imaging, lymph nodes assessment, and biopsy of metastatic sites. This to confirm distant spread and to guide decision about systemic therapy, including immunotherapy or targeted therapy. treatment of melanoma really differ depending on the stage. So if patients are on early stage disease ranging between 0 and 2, the treatment is dominated by surgery, mostly wide local excision. This is often combined with sentinel lymph node biopsy in those higher risk cases. So for patients with early stage melanoma, the 5-year survival ranges about 70 to 95%, and this depends on the stage and risk factor. However, up to 30% of patients, this can still relapse, and especially those people that are like in the higher risk stage 2 group. When we get to the late stage disease, so stage 3 and 4, the treatment landscape is quite different. We now have access to 3 major systemic options, which is immunotherapy, targeted therapy, and chemotherapy. And among the 3, immunotherapy has become the most popular option and have revolutionized the melanoma treatment. So if you look at the survival from advanced melanoma, the 5-year survival rate has improved from less than 10% around 2011 to more than 55% in 2024. And this is in the adjuvant setting, so when the treatment is administered after the surgery. However, in the recent years, a new approach has been established called neoadjuvant setting, where the treatment is given prior to the surgery. And this has given us an improvement in the 5-year survival up to 75%. However, even if the survival rates for many advanced melanoma patients have increased drastically, we still have a high percentage of patients that do not respond to the therapy and eventually relapse. In addition, as I showed you earlier, in the early stages, a subset of patients still progress despite apparently low-risk primaries. And so there is a critical need for biomarkers, and we need to know which patient will respond to the therapy or relapse, and now we can intervene earlier and more precisely. So in this slide, I try to summarize the unmet needs for biomarkers in both early and late stage melanoma disease. In particular, in early stage disease, we still rely heavily on histopathology and clinical judgment, which could be subjective at time and of course can limit the accurate risk stratification. Conventional pathology, on the other side, while incredibly powerful, still doesn't capture the biology and the immune context that might predict which tumor will progress. So for this reason, we need an improvement of these approaches through molecular immune biomarkers that could be potentially from both tissues and blood to distinguish low from high-risk lesions. But also, in addition to that, through the development of this new technology, spatial and transcriptomic profilings that will help us to capture the early tumor-immune interaction which are linked to progression. And today I'm going to show you some of this data that we have developed in our lab. On the other side, during the late-stage disease, single-site biopsy and imaging typically fail to capture tumor and immune heterogeneity. And also the current staging doesn't really help us to predict response or resistance to immunotherapy. And for this reason, we're trying to apply multi-omic and spatial biomarkers to resolve the functional and tumor immune states within the metastasis. And additionally, trying to understand whether we can develop dynamic biomarkers to monitor immune adaptation and emerging resistance during treatment. The core research of our laboratory is the tumor immune microenvironment, or TME. So on this slide, you can see some of the key features of the immune players that can be present within the melanoma tumors. So on the positive side, we have cells such as the cytotoxic T lymphocyte, or CD8 T cells, and Th1 polarized CD4 T cells, which are associated with effective anti-tumor immunity. On the negative side, as you can see down here, we have regulatory CD4 T cells, myeloid-derived suppressor cells, and M2 polarized macrophages, all of which can suppress effective immune responses. So understanding the feature of the TME in different patients can be a powerful tool to predict melanoma development or treatment response. I'll start with our work in late-stage disease, which has been very recently accepted in Nature Communication journal. So the study specifically focuses on lymph node metastases from melanoma patients that were treated with immune checkpoint inhibitors, and we analyzed the TME of this patient to understand the spatial cues to predict immunotherapy response. In this cohort, we collected lymph node metastases from 32 patients, and we grouped them into three main categories: untreated patients, patients who were resistant or responders to immune checkpoint blockade. We then applied a multi-omic and spatial approach. So we got both FFPE, so formalin-fixed paraffin-embedded blocks, or fresh tissues from these patients. And so from the FFPE, we perform multiplex immunohistochemistry to visualize multiple markers in situ, or nanostring assays to profile the gene expression from immune panels. On the other side, through the fresh tissue samples, we perform flow cytometry to look at the cell phenotypes, bulk RNA sequencing to capture gene expression at the tissue level, and single-cell RNA and TCR sequencing to define cellular states and clonotypes at a high resolution. On these slides, you can see the workflow for the bulk RNA sequencing. So we start from the FFP blocks from the lymph node biopsies. We section these into very thin tissue slices, extracted the RNA, created the RNA libraries, and then sequenced the RNA to capture the transcriptional profile of these cells. And so what we observed is that comparing the responders and the resistant patients, we look at the differentially expressed genes and pathway scores. We observe that responders show a higher expression of genes linked to myeloid activation and antigen presentation, as you can see on the right side of the graph, and higher score for cytotoxic CD8 T cells, dendritic cell type 2, and interferon-activated tumor-associated macrophages, which are really important for anti-tumoral immune responses. In the gene set enrichment analysis, responders show enrichment pathways that are related to T-cell proliferation activations, natural killer activation, and cellular response to interferon gamma. By contrast, resistant patients showed enrichment of pathways associated with melanoma metastases, G2M, cell cycle, hypoxia, and relapse signatures. So even at the bulk level, the lymph node metastases of responders look more immunologically hot, with strong cytotoxic and antigen presentation programs, where the resistant lesions show a feature of proliferation, hypoxia, and aggressive tumor programs. To go deeper with the analysis, we then use single-cell RNA sequencing, and as shown in these slides, we started with fresh lymph node biopsies. We digested the tissue and purified the individual cells, which were then we extracted the RNA from the single cells and then performed the RNA sequencing. These allowed us to profile the transcriptome of each cell individually, and it gave us much more granular view of the cellular diversity within these metastases. Here we show the UMAP plot for our single-cell RNA-seq, and as you can see, the full landscape of cell type that we capture. So we have the green large cluster representing the T cells, the brown clusters here representing the melanoma cells, the purple one, myeloid cells, the red one containing the B cells, and then you also can see we also analyze endothelial, fibroblast, NK, and proliferating cells. So this gives you a sense of the cellular complexity within these metastatic lymph nodes, which is just not tumor and T cells, but it's a rich ecosystem of immune and stromal cells all interacting within the tumor microenvironment. So using the UMAP, we were able then to further analyze and subcategorize the T cells clusters. So we identified, as you can see here on the left side, 15 these different clusters of CD8 T cells. And these clusters included naive CD8, lymph node-associated effector T cells and tumor-resident CD8 T cells up here. For the CD4, we identify cluster representing various T regulatory subset, naive and central memory T cells, Th1-like cells, follicular helper-like, and a distinct, which is this number 11 here, distinct resident memory-like clusters. So what are these tumor-resident memory T cells and why do we care? So TRMs are defined by their ability to reside long-term in tissues without recirculating. Phenotypically, they are identified by the expression of markers of tissue retention such as CD49A, CD103, and CD69, alongside with inhibitor receptors such as PD1 and CTLA4. Often, high levels of effector molecules such as interferon gamma, granzyme, and perforin are typical of these cell populations. So these cells have been studied and they're poised for rapid local responses when they encounter the antigen. And as you can see also that they've been demonstrated that TRM can interact directly with melanoma cells, releasing these effector molecules to mediate tumor cell killing. Given the importance of these cells in regulating anti-tumor response against melanoma, we first asked whether the TRM gene signature was enriched in the lymph node metastases of our cohort of patients. And using the nanostring technique, we calculated this TRM score based on a panel of transcription factors, inhibitory receptor, and phenotype and tissue retention genes. And what we found strikingly was that responder patients to immune checkpoint inhibitors had the statistically significant higher score for TRMs in these patients. We then moved to multiplex immunohistochemistry to quantify the tissue-resident memory T cells in space and understanding their location within the tissue architecture. As shown here, we first used the H&E staining, the traditional histopathology approach, to identify tumor regions versus normal lymphoid tissues. And this was critical because the pathologist expertise allowed us to precisely delineate where the metastatic melanoma is located within the lymph node tissue, and this guide our subsequent analysis with the multiplex immunohistochemistry. So on the right side here, you can see the MHSC panel where we simultaneously visualize different markers. So we have the DABI for staining the nuclei, SOX10 to mark the melanoma cells, the CD3 in red, the CD4 and CD8 markers, CD69 as tissue resident marker in green, CD103 is as another tissue resident marker in white, and TCF1 to identify stem-like progenitor of T cell states. So by combining pathologist annotations of the tumor versus the normal tissue with this multiplex staining, we can precisely quantify where the CD8 and CD4 tissue-resident T cells are located. And what we observed was that when we quantify these CD8 TRT cells, we found that specifically in the tumor area of the lymph nodes, responder has significantly higher density of both TCF1 positive and TCF1 negative TR compared to the untreated and resistant patient here on the right side. So this distinction is quite important, especially because TCF1 positive TR cells represent a more stem-like or progenitor state with self-renewal capacity and the potential to generate effector progeny. While on this side here, the TCF1 negative TR cells are typically more terminally differentiated effector with immediate cytotoxic function. And the fact that responders showed enrichment of both populations suggests that they maintain a balanced T cell response. And importantly, in normal lymphoid lesion areas, there was no significant difference between the two groups, suggesting that their enrichment is particularly within the tumor region where these T cells encounter melanoma antigens. While multiplex immunohistochemistry allowed us to quantify these TR cell populations spatially. Single-cell RNA sequencing enabled us to define their molecular state in much greater detail. As shown on this slide, we zoomed on the CD8 T cell compartment and identified different subclusters of CD8 T cells within the lymph node metastasis. And as you can see here on the left side of the slide, the UMA plot shows this cluster color-coded by their identities. So here on the right side, we can see the CD8 naive T cells. In the central area, we have the lymph node-associated CD8 effector T cells, and here on the upper right side, the tumor-resident T cells. And to validate that these tumor-resident were actually real, we applied a module of gene that including all the features for TR cells. And as you can see here in red, these 6 clusters had very high expression of this TRM module. So this molecular diversity reveals that tumor-resident T cells are not a single homogeneous population, but rather comprise multiple functional states, some more cytotoxic, some more proliferative, some expressing chemokines to recruit other immune cells. And so understanding this heterogeneity is critical for predicting which patient could respond to immunotherapy. We then characterize the function of these CD8 TR T cells, particularly their cytotoxic capacity. So in the left panel here, we displayed a dot plot showing the expression of interferon gamma across the different CD8 T cell clusters. And as you can see, as highlighted in the pink square, the TR clusters are the ones that had the highest expression of interferon gamma, meaning that not only these T cells are highly abundant in the TME of responder patient, but also actively produce interferon gamma, which is a key mediator of anti-tumor immunity. Here on the right side of the slide, we then perform protein level validation using flow cytometry. So in the top graph, you can see the TR T cells that are like antigen specific, so expressing CD39, but also this T cell expressing granzyme B, which is a key cytokines for cytotoxic function. And so here we can quantify the these granzyme positive antigen specific CD8 TR cells, and as you can see, the responder patients to immune checkpoint inhibitor had the highest frequency of these cells. On the right side, we actually also show the functional degranulation assays of granzyme B, where we co-culture tumor-infiltrated lymphocytes with melanoma cells, and we quantify the activity of these TR T cells to degranulate in presence of melanoma cells. And as you can see, just the co-culture with the autologous TILs and the melanoma cells increased drastically the degranulation activity, meaning that the CD8 TR T cells respond to the antigen specifically and they start degranulating granzyme B to then potentially kill the tumor cells. To validate this cytotoxic capacity, we then ask whether these TR population were clonally expanded. So we perform TCR sequencing and trying to understand and track the clonal diversity and expansion part of these T cells within the lymph node metastases. So as is shown here in the slides, this is a schematic that illustrates how TCR sequencing works and why it is important for the clonal expansion. So on the top side, we have single-turn small clones, meaning that these T cells usually do not respond to any antigen, so they do not expand. But when the T cells, through the TCR, recognize the antigen on the tumor cells, these are starting expanding exponentially. And so we were able to then extract the RNA and create the library from these TCR sequences and then sequence the data. So this allows us to determine the clonal expansion of specific T cells compartments. So we then applied the TCR sequencing in our cohort of patients, and what we found is that the granzyme-positive TRT cells, so those cytotoxic tissue-resident cells we've been discussing, especially in the responder patient, as you can see here, they displayed hyper-expanded TCR clones. So this is consistent with sustained antigen-driven responses where these T cells have recognized melanoma antigens, proliferated extensively, and maintained their presence in the tumor microenvironment. So in other words, these TR cytotoxic cells in responders are not just abundant and functional, but they also clonally focus on tumor antigens, so which likely contribute to controlling melanoma growth and driving responses to immune checkpoint inhibitors. Apart the CD8 T cell compartment, we hypothesized that CD4 T cells could have a role in sustaining immunotherapy response. And so for these reasons, we analyzed the CD4 T cells from the same lymph node metastasis across all patients. And as you can see here, we identified a CD4 TR clusters highlighted in pink, which had a very high TR score module. On the right side of the slide, we then explore whether the abundance of this population was enriched in one of our patient group. And as you can see here, as for the CD8 TR cells, the antigen-experienced CD4 TR cells, so those expressing CD39, CD69, and CD103, were significantly more frequent in the responder patients. So in addition to total abundance, we then use multiplex immunohistochemistry technique to see the spatial distribution of these cells within the tissue section. And so in the pink highlighted box shows here that CD4 TR cells density in the tumor region of the lymph node metastasis of responder patients were highly increased in the responders compared to the resistant. And importantly, in normal lymphoid tissue area on the left side of this graph, there are no significant differences between the groups, which mirrors what we already observed in the CD8 TR cells. So this tells us that the enrichment of CD4 TR cells is specifically within the tumor microenvironment where they encounter melanoma antigens and could contribute to effective anti-tumor immunity. We then characterized the functional status of these CD4 TR T cells, particularly their cytotoxic capacity. And as you can see on the left side of the slides, we displayed the interferon gamma expressions, and as you can see, these TR T cells here had the highest cytotoxic modules combined with the highest expression of interferon gamma. This tells us that these CD4 TR cells, like their CD8 counterpart, are actively producing interferon gamma and contributing potentially to anti-tumor immunity. And so to further validate these, we then look at the antigen experienced TR-T cells expression and those expressing also granzyme B. So as you can see on the top right corner, we analyze the abundance of granzyme positive antigen experienced CD4 TR cells across the different patient groups. And again, similar to the CD8, we have the CD4 TR highly enriched in the responder patients. On the lower side of these slides, we then show the function of the granulation assay, where we co-culture again the tumor infiltrating lymphocyte alongside with the autologous melanoma cells of each patient. And we measure the CD107A expression as a marker of degranulation and cytotoxic activity. So here again, the bar graph displays the CD107A expression, specifically within the CD4 TR compartment. And once again, these cells, specifically in the responder patient, started degranulating upon antigen recognition, and that happened just in the responder patient. So these basically demonstrate the CD4 TR cells in responders are not just helper cells, they're also directly cytotoxic and potentially capable of killing melanoma cells. Finally, I wanted to show that these CD8 TR-T cell subset could have predictive potential across an independent cohort. At the top of this slide, you can see the study design. So we have two cohorts of melanoma patients with lymph node metastases. The first on the top is our cohort. The second is an external cohort that we obtained from Dr. Ramona Schlenker and colleagues, which included 11 untreated patients. On the bottom left of this slide, we projected the CD8 T cells from the Schlenker cohort onto our reference map, so the human plot that we generated from our own single-cell RNA sequencing. And here on the right side, you can see the bar graph showing the frequency of the different CD8 T cell clusters of the external cohort. So what we can observe is that specifically cluster 10 to 13, which corresponds to some of the TR clusters from our cohort, are highly enriched in this external cohort as well. And so this validates that these TR states are not unique to our cohort, but are a generalizable feature of melanoma lymph node metastases. Here on the right side, basically what we showed is that the patient follow-up data from the untreated cohort, where this patient eventually received immunotherapy and became either resistant, so as you can see here in pink, or responders. And here we track the proportion of the different CD8 T cell subset in resistant patients versus responders. And the key finding is highlighted here in green is that granzyme positive TR cells and NKG7 TR cells, which are the most cytotoxic TR cells, show significantly higher proportion in responders. And so this cross-cohort validation is critical because it demonstrates that the association between cytotoxic CD8 TR cell states and immunotherapy response is reproducible and potentially predictive across independent patient cohorts. In addition, it suggests that TR subset, such as this cytotoxic one, could be potentially serve as a biomarker to predict which patient will respond to immune checkpoint inhibitors. So far, I've focused on whether TR cells are abundant in responders and what they look like transcriptionally and functionally. But the next question is whether they are located within the TME and who are they interacting with. So from previous studies, we know that in normal lymph nodes, dendritic cells, particularly dendritic cell type 1, interact with CD8 cytotoxic T cells and CD4 follicular helper T cells via the MHC class 1 and 2 and T cell receptor interaction, in addition to co-stimulatory molecules such as CD40 ligand and CD40 and the production of cytokines such as interleukin 4 and 21. And so this interaction helps to prime effective immune responses. However, it is not clear what happens in lymph node metastases, especially in your response in melanoma. So given the observed abundance of both CD8 and CD4 TR cells in the tumor area of responder patients, we hypothesized that a similar interaction of these TR cells could happen with the dendritic cells. And then we hypothesized that these triads are fundamental to induce the cytotoxic function of TR cells to then target melanoma cells using granzyme, interferon gamma, and perforin molecules. So using multiplex IHC analysis and spatial analysis with the HALO software, we identified what we call immune triads in lymph node metastases, as you can see here. So these are microregions where we identified dendritic cells in purple, CD8 TR cells in green, and a CD4 TR cell in blue, which are in close proximity within about 30 microns of each other. And then we quantify these triads in responders and resistant samples with CD8 TR and CD4 TR cells in close contact with dendritic cells within the tumor regions. And so proximity analysis show that these triads are significantly higher and frequent in responder patients, as you can see here, but they are completely absent in resistant patients. And this specifically happens within the tumor microenvironment. So conceptually, these triads represent a spatially organized unit of effective anti-tumor immunity. So the dendritic cells presenting tumor antigen to the CD4 and CD8 TR, which potentially execute cytotoxic killing, all in a tightly connected microenvironment. And so this led us to propose a spatial biomarker models. So in lymph node metastases, as you can see here, the presence and density of these triads could serve as a spatial biomarker of immune checkpoint inhibitor response. So in other words, we may be able to predict response not just counting cells or measuring gene expression, but also quantified specific spatial relationship between these immune cell subset and melanoma cells. So after showing these preliminary results on the late-stage disease, I would like to show our work that we did on the early stage, where we asked two critical questions. So, do tumor-resident T cells have a role in the progression of early-stage disease, and whether the markers that identify these cells can function as a biomarker of relapse? As I mentioned earlier, while we've made tremendous progress in treating advanced melanoma with immunotherapy, up to 30% of patients with early-stage disease still relapse after surgery. So we do need better biomarkers to identify these high-risk patients at the time of diagnosis. So in our early-stage cohort, we focus on stage 1A and B melanoma patients. And so these are patients with relatively thin primary tumors who are often considered low risk. And the study design is shown here. So all the samples for this project were collected at baseline, which is the first biopsy before any treatment. And so a patient underwent surgical resection by a dermatologist or a GP, and the tissues were collected. These patients were then followed up over time and classified them into three main groups based on the clinical outcome. So the first group up here, the non-recurrent patients, are those patients who remain disease-free after surgery. The middle group here developed recurrence within 2 years post-resection, and the bottom group here developed recurrence within 5 years post-resection. So the crucial point here is that we're looking at biomarkers in the primary tumor at the time of diagnosis and asking whether those baseline features can predict who will relapse down the line. And so this is the key to developing a clinically useful prognostic test. Our experimental approach for studying early-stage melanoma parallels what we did in the lymph node metastases, but it was adapted specifically for primary tumors. So here, the only available tissue, it was from FFPE blocks. And what we did, we started with H&E analysis, so it's standard histopathology, where our pathologists identify the tumor regions versus stroma in the primary melanoma samples. Then this pathologist annotation was absolutely critical because it allowed us to precisely map where the melanoma cells are located versus the surrounding stroma tissue. From here, we had two complementary approaches that fed into each other, showing the spatial relationship. So what we have here on the right side, we perform gene profiling using nanostring immune gene expression profile, which gave us the quantitative data on immune cell expression signatures across different regions of the tumor. And here on the bottom side, we perform multiplex immunohistochemistry for tumor resident localization. And so this is the spatial component where we visualize multiple markers simultaneously. So the key innovation here is that integrating the spatial relationships where the TR cells are located and with the gene profiling data. And so this integrated approach allowed us to ask whether the TR cells are present, where they are located, but also their functional state. And so here, starting with the multiplex immunohistochemistry, we use this same panel that I showed you earlier for the lymph node metastases. And so we use these 7 markers to identify T cells and different states of T cells. And this marker here, SOX10, to identify melanoma cells. When we first look at the overall proportion of T cells in these early stage melanomas, comparing non-recurrent patients in green, the 2-year recurrent in pink, and the 5-year recurrent in red, we didn't see dramatic differences. So here on the left, you can see the individual patient samples, and on the right, the summary box plots. So the proportion of total T cells as percentage of total cells were similar across the three groups with no statistical significance differences. And so this tells us something really important, which is not just simply how many T cells are present overall. So we need to look deeper at the specific T cell subset and where they are located. And so this is where the tumor-resident T cells become particularly interesting, because they represent a functionally distinct population that may be critical for local immune surveillance and preventing tumor recurrence. So when we specifically examined the CD8 tumor-resident T cell subsets, we saw striking differences that were not apparent when looking at the total T cells. So this slide shows the density of the CD8 TR cells across the three different group patients, and the data were separated into the stroma on the left side and the tumor area on the right, where we measured two main populations. And so overall, TCF1 negative and positive CD8 TR cells in non-recurrent patients had significantly higher density compared to those patients that relapsed. So this tells us that CD8 tumor-resident T cells in the TME, particularly within the melanoma TME, are associated with protection against relapse. And so patients who remain disease-free have more of these cells present at baseline. We then observed a similar pattern for the CD4 tumor-resident T cells, and again, we measured the CD4 TR density in both stroma and tumor compartments, looking overall at the TCF1 negative and TCF1 positive CD4 TR cells. And these mirror what we saw for the CD8 TR cells. So non-recurrent patients are characterized by higher density of these cells within the tumor. And so both CD4 and CD8 compartment might contribute to this protective effect. To further explore this relationship, we then perform single marker analysis focusing on CD103 as a TR marker and SOX10 as a melanoma cell marker. So here on the left side of the slide, we calculated the ratio of CD103 to SOX10 expression, so essentially the ratio of TR cells to melanoma cells, shown here in a log scale. And again, in the tumor compartment, you can see that non-recurrent patients in green have significantly higher CD103 to SOX10 ratio compared to 2 years and 5 year recurrent patients. Here on the right, we then perform correlation analysis between the mean of CD103 and SOX10 expression, separated again between stroma and tumor. And in the tumor compartment, as you can see, we have significantly positive correlation for non-recurrent patients, meaning that patients who don't relapse, as melanoma cell increase, TRM increased proportionally, suggesting an active immune engagement with the tumor. So by contrast, the recurrent group show no significant correlation, suggesting that in this patient, the TR response is not keeping pace with the tumor. And so this analysis reinforced our finding that higher intratumoral CD103 TR density at baseline is associated with protection against relapse. We then validated our results of the primary melanomas on a second cohort of stage 2 melanoma patients, and this work was done in collaboration with Bristol Myers Squibb. So here in the box plot, you can see the 4 different TR cells that we studied in our research. So on the top, we have the TCF1 negative and positive CD4 TR, and in the bottom, the TCF negative and positive CD8 TR. And as you can see, specifically the TCF1 negative CD4 TR were enriched in the tumor areas of non-recurrent patients and both CD8 TR population as well. So from a translational perspective, this is particularly exciting because multiplex IHC is becoming increasingly implementable in routine clinical pathology laboratories. And so these markers that we use in our project are also well-characterized antibodies that can be integrated into diagnostic panels. So this means that we're not talking about a research-only technique, but a potential clinical tool that could stratify relapse and guide treatment decision in early-stage patients. So to summarize, we show that tumor-resident T cells, both CD4 and CD8, are significantly enriched in the tumors of patients who do not relapse after surgical resection. So this enrichment is mostly pronounced within the tumor compartment rather than surrounding stroma, suggesting that these cells are actively engaged in immune surveillance despite showing marker of chronic activation. And so these findings echo what we saw also in the late-stage disease. So just as responder lymph node metastases are enriched for cytotoxic TR cells and immune triads, non-recurrent early-stage tumors are enriched for intratumoral TR cells. And so this suggests a shared protective role for tumor-resident T cells across the melanoma disease spectrum. And so from a translational perspective, TR density could form the basis of a biomarker assay to stratify relapse risk in early-stage melanoma and IO response in late stage. And so this could guide decision about surveillance intensity, adjuvant therapy, and patient counseling. In addition, the beauty of this approach is that it's based on technologies such as the multiplex immunohistochemistry and spatial analysis that are becoming increasingly available in clinical pathology laboratories. And so we're not talking about research-only techniques, but these tools can be realistically implemented in routine diagnostic workflows to improve patient care. Before I end this presentation, I would like to take a moment and acknowledge the many people and organizations that made this work possible. First and foremost, I would like to thank my supervisor, Professor Paul Neeson, and my dear colleague, Dr. Louise Howe, for their guidance and support throughout this research. To the past and current members of the Neeson Lab and our clinical collaborator, Peter Mack, the Melanoma Research Victoria, and the Medical Oncology Unit. This work was done in collaboration with industry partners at Roche and international collaborators such as Mitch Levesque at the University of Zurich and Daniela Thomen at the Netherlands Cancer Institute. Last but not least, I would like to acknowledge our consumer advisors who provided invaluable patient perspective, and most importantly, I would like to thank the patient and their family who participated in this research. None of this work would have been possible without your generosity and willingness to contribute to science. And thank you all for your attention. Happy to answer any questions.
Thank you very much, Andrea, for that wonderful presentation. We will now begin the Q&A session. And for the audience, please make sure to submit your questions for Andrea using the Q&A system in the live stream player. We already have a few questions, so let's begin. The first question is, what are the key steps required for translating these spatial biomarkers into clinically validated assays that could be used for routine pathology? And are there specific challenges in terms of standardization or cutoff thresholds?
It's a bit a complicated question in a way that they need several steps. So the first is, of course, validating that to bigger cohorts and, of course, external cohort from our institute as well. So that's what we, as I showed in the last slide, we're trying to expand with international collaborators, so to expand this approach and see whether we could validate our results. And in terms of implementation to the pathology laboratory, so there are like two main stream of works. One is the basic from single marker analysis. So in our case, could be, for example, an antibody with CD103, and that can be applied on H&E slides. And so we could, for early stage patients, potentially it can help to predict whether this patient relapse and maybe needed more insight over time compared to those potentially that had very high signature, and so they will remain tumor-free. In addition to this, what we're doing in the lab in collaboration with different computational biology groups is that
we are starting also to do a high resolution H&E scanning and apply AI signatures and machine learning on these samples. Because ideally what we could do is, yes, we can stain with a single marker, so the CD103, for example, but that can feed into the algorithm and potentially increase the predictive analysis of our results. And so that potentially I can envision in the next 5 years with the AI, not just as we see already as chatbots, but in terms of it would be super powerful as a tool for predicting the results and on the imaging side, of course.
Thank you. And the next question is, are there any specific insights around the mechanistic role of the triads? Are they just markers of an effective immune response, or do they play an actual causal role in anti-tumor immunity?
So this is a new project that is budding from the publication that has just been accepted. So I have a new PhD student that is going to start on the mechanistic insight. So we have another collaborator across the street, actually, from our building, with an immunologist that has mouse models for melanoma lymph node metastases. And so given the insight, because what we had, of course, is a snapshot analysis. So we got these patient samples at a certain point in time, but we cannot, of course, track how the immune response to the triad formation is developed. But fortunately, we can go backwards. So actually, in our lab, it's very peculiar because usually most of labs in the world, they start from mouse studies and they trying to translate towards human. We are doing backwards. So we have all this information and powerful information from real human patients, and we're going backwards into a mouse model to validate what we observed in humans. So potentially in the next couple of years, of course, we're going to have results on how these triads are formed and why they're so important for anti-tumor response.
Thank you. And the next question, you described DC, CD4 TR and CD8 TR immune triads in lymph node metastases. So the question is in regards to that, have you identified analogous microstructures in primary tumors? And if not, what do you think explains the difference?
This is a really good question, actually. So yeah, we are literally starting next week to look this triad into the early stage patients. So we just received a very powerful new microscope in our institute that allows us to actually up to 40 different markers at one go. And so that will allow us not just to show these triads, but also potentially the different dendritic cell type all in one go. So yeah, very soon we will have the results for that too.
Thank you. And we have time for one more question. The last question is in regards to the finding that exhausted or terminally differentiated CD8 positive TRM cells expressing high levels of inhibitory receptors. Is this actually associated with better outcomes in early and late stage disease? There seems to be a paradox there.
Yeah, so because the exhausted T cells have been established initially in the context of chronic infection, of course, in a mouse model. And so what's happening now is that we are trying to translate what has been studied actually the name of, in this presentation, I call these cells tumor-resident T cells rather than tissue-resident memory T cells, which were the original phenotype for these cells, is that apparently what happens in tumors, so these cells, even if they express some markers of exhaustion, they're still very active. And actually, that we found that these are not only active, but they are like tumor specific. So, for example, as I show you in one of the slides, is that the majority of these cells actually had the expanded TCR, which means that they recognize the antigen and they clonally expand to respond to the tumor as well. So yeah, it's a new concept. Like there is a lot of talk between immunologists about how to identify these or rename these cells. And so that's why for this last paper that we published, we landed on tumor resident. So they just make sure that they're like within the tumor microenvironment.
Wonderful. Thank you so much, Andrea. We'll move on now to our next session. And I'm pleased to welcome Tony Bahinsky. Tony Bahinsky's career spans academic research and large pharma, with more than 25 years of experience in the pharmaceutical industry. He served on the advanced technology team at Harvard's Wyss Institute, leading DARPA and FDA collaborative efforts in the development of organ chip systems. He had also served on several advisory boards and is currently a member of the Science Board of the US FDA and formerly the US EPA Board of Scientific Counselors. He is a member of the editorial board of the journal Applied In Vitro Toxicology. Tony is the author and co-author of over 45 publications, including peer-reviewed articles and book chapters. Warm welcome to you, Tony. The floor is yours.
Thank you for the introduction. I'd like to talk today about using high throughput automation and human relevant tissues for biomarker discovery. As we all know, drugs entering clinical trials, only 1 out of 10 actually succeed. And there's a great need for better translational models. Early in the discovery process, compounds are screened against cells in a dish. Gives them very low data, but gate points to move forward on to better translational models such as animal models. And these are used to predict the human responses. The problem is that the animal models aren't very predictive of either efficacy or safety. And so once the compounds get to the clinic, anywhere from $50 to $100 million to $1 billion can be spent in phase 3 clinical trials just to see these drugs and therapeutics fail. The total cost per year is around $90 billion a year. And the main reason is because human outcomes are very different from animal outcomes. Based on this, there's been recent US policy and regulatory encouragement by the FDA to use novel alternative methods. These include organs on chips, microphysiological systems, computational tools such as AI, and to be used for preclinical drug and biosimilar applications. The hope is that these human-based systems can predict and are much more translational than either the in vitro preclinical cells in a dish or the animal models. And there's been a paradigm shift within the FDA. They recently released a roadmap to reducing animal testing and preclinical safety studies. And this has been endorsed by the new FDA Commissioner, Martin McCary. And there's a large effort across the industry, including the NIH, pharma, to incorporate a lot of these complex in vitro models and novel alternative methods, and hoping to refine the drug development process to make it better, more translational, and derive better safety and efficacy for patients. Now, the current alternatives to animal models really aren't efficient. As I mentioned earlier, 2D assays are not very realistic. You can do them at high throughput, but it really limits the predictivity, and especially if you want to utilize that data for modern in silico modeling, such as artificial intelligence or machine learning. Organoids are still simple, they're very inconsistent. The generation of these is highly variable, and there's still a degree of immaturity within the tissues developed by these organoids. So they're not great translational models. Moving to existing organ chip models, so these have great promise. They started in 2010 with a seminal publication by Dan Ha in the lab of Don Ingber at the Wyss Institute. and really kickstarted the organ-on-chip systems field. Looking at human breathing lung on a chip, they were able to recreate much of the physiology and the pharmacology in that organ system. Since then, there's been a lot of different companies and a lot of academic groups developing organ chips for multiple different types of tissues and for various indications such as safety and or efficacy. The problem is that they're still very manual labor intensive. And due to that, there's still large variability. They're unscalable. You can do very large numbers. So their utility across the broad spectrum of drug discovery is really limited to the end stages such as lead discovery or looking at compound optimization. And because of the manual nature, a lot of these systems, they tend to be very irreproducible. Now, we've really tried to solve these issues by developing a fully automated end-to-end platform that directly tests, establishes, cultures 3D vascularized human organ tissues. We've done various different complex human tissues, and they're similar to lab-grown biopsies. If you look on the left of your screen here, there's a tissue that looks like a pencil. It is basically a vascularized bone marrow tissue. It's about 1 centimeter in length and about 0.5 centimeter in width and height. Now, we've utilized these microphysiological systems and scaled those so that we can do massively amounts of these, thousands to 10,000s at a time. We've generated customized discs, and then we've generated robotic systems to reproducibly generate, culture, and then interrogate these tissues at scale. And these systems are called hives. It's a fully automated platform that has a robotic arm. It has a self-contained confocal 3D microscope, and we can also do live imaging. The capacity is anywhere from 1,000 to 10,000 different independent tissues, self-organized from tissue-specific primary human cells. We can independently dose, sample effluent from the tissues. We can then fix and stain at the end of a study and do high-resolution imaging. The automation allows us to do operations unattended for weeks. So we can not only do acute studies, but the nature of these, as you will see in the remainder of the talk, we can culture these for multiple weeks and actually do semi-chronic or chronic type studies on these tissues. Now, each tissue is made from over 200,000 patient-derived cells and is indistinguishably realistic and functional. We've looked at different modalities. We've made lung cancer tumoroids that are vascularized tissues. We've done respiratory tissues that include lung macrophages and the innate immunity. And we've also done liver and looked at disease states like lung fibrosis. Now, we utilize tissue scale self-assembly to create these human primary tissues. We put a mix of collagen, ECM, and the cells into a center channel, and then vascular cells lining the outside channels. And over the course of about 7 days, we get vascular formation inside the channel and anastomoses to the side channels so that we now have fully perfused vascular tissue. This tissue self-assembly really recreates realism across the nanoscale to the macro scale. As you can see here, it is an image of our vascular network. The endothelial cells are stained with green and CD31, and then the stromal cells surrounding it are stained with phalloidin. On the right there is an ECM or electron micrograph, a standing electron micrograph. And you would be hard pressed to distinguish this from an SEM of native vascular cell. We see lots of the key characteristics that you would want to see in a vascular network. In the lower left corner, you can see this sort of handshake between two cells that causes the tight junction between endothelial cells and the vascular barrier. Now we rely on self-assembly, and this is a time-lapse video over the course of about 5 to 7 days, which shows the red cells that are labeled, the CD31 labeled vascular endothelial cells, and then in green are endothelial cells that are labeled GFP on the outsides. And over the course of 7 days, you can see that the vascular cells congregate, form the vascular network in the center channel, and then anastomose with the side channels to allow a fully perfusible network. The tissues are dynamically perfusible. In this case, we're perfusing the vascular network with human red blood cells, but we typically perfuse the cells with media that is appropriate for the various different organ tissue types. I'll give an example of how we utilize these tissues utilizing our bone marrow tissue. With the bone marrow, we seed with CD34 positive hematopoietic stem cells, and over the course of about 9 days, the stem cells differentiate into the vascular hematopoietic niche along with a perfusible vasculature. We have vessels, neutrophils, macrophages, among other different various cell lineages, and they mature over time with the model cultivation. The beauty here is that we do not have to 3D print. We don't have to localize the cells in any manner. The cells know how to self-organize themselves, and the biology really drives the formation of these highly organized and highly translatable human tissues. This just gives a little bit more of a schematic over the time course of the establishment of the bone marrow niche. As I said earlier, we have the endothelial cells seated on the side channels, endothelial and stromal cells, including the CD34 hemolytic stem cells seated in the center channel. Over time, the endothelialized lumens form on the side channels. The established niche forms, the bone marrow niche forms in the center channel, along with the vasculature, which then anastomoses to the side channels. And you can see the bright field images on the bottom here as it forms, and then the stained image on the bottom right. We get a rich lineage of marrow with about 400,000 cells per tissue. This shows a little higher resolution imaging of the bone marrow niche itself. And over the course of several weeks, we get differentiation of the hemolytic stem cells from these 34 all to the mature cell types. We have maintained proliferative cells within the niche so that to replenish the different various progenitor types. We get granulopoiesis and then maturity into CD16 positive macrophages. In addition, we have erythropoiesis, CD71 and CD235A positive cells, in addition to development of blood monocytes. So we have a rich niche. When we do a little higher resolution interrogation of the phenotype that we see, we can see in panel K, we have nurse macrophages along with erythroid cells causing these islets, which are characteristic of the hematopoietic niche in the bone marrow, the human bone marrow. The vascular endothelium lumen and the association of granulocytes with that, as well as the development and the maturation of neutrophils in O and P panels. We can take these tissues and we can dissociate them either by removing the tissues or dissociating them in situ and start to do transcriptomic analysis of the engineered bone marrow. And as you can see here, we have all the different populations that are formed. We have the myeloid cells populations, erythroblasts, eosinophils, the perivascular stromal cells, as well as the endothelial cells. And we can capture the trajectories of how these cells are maturing for each one of these cell fates. We can do pseudotime analysis, which is a snapshot in time, but shows that we have the different stages of each one of these populations with the key critical markers showing that are differentiating over time into the multiple different lineages that are present in human mature bone marrow. This also indicates that we have continuous turnover of progenitors into mature cells over the time course of these studies at the vascular niche, and that rather than a single pulse of differentiation, which then dies off. We can do very sophisticated receptor ligand interaction analysis. This is where we can utilize transcriptomics to look at the receptors present on one cell type and the ligands present on another cell type, and then analyze how those two are interacting, seeing which receptors are present on, say, the endothelial cells, and then interacting potentially with a fibroblast cell. Which pathways are present, which ones can we interrogate, which can lead to, which receptor pairs we can utilize to generate potential biomarkers for those pathways. We not only see and interrogate the cells that are present inside the tissue, but we can also sample the effluents that are coming out from each one of these tissues. One example here is we can look at neutrophil mobilization. This normally occurs in healthy bone marrow. And so the neutrophils are matured in the bone marrow, they egress out into the vasculature, and then we can collect those and identify and quantitate how many of those are leaving over time. One of the things I highlighted was the irreproducibility and the variation, inherent variation with doing manual tissue development or manual organ-on-chip work. Here, this, I think, really kind of drives home how the automation drives high consistency. So this is a snapshot of a study we did with cisplatin to look at the toxicity of this chemotherapeutic agent, this oncology agent, on the bone marrow, which is a classic toxicity. And we have the healthy control in the first column. If you look down, there are 9 replicates. These are all individual tissues. So this is a look through an amalgam of the 3D tissue in 2D. And so there's really high consistency across each one of these individual tissues. They're not slices of a single tissue. These are all technical replicates. And then even when we start to add in cisplatin, which is the toxic agent, you can see the consistency still is maintained from top to bottom across each one of these indications. And so it really hits home, that you can make subtle changes or identify subtle toxicities or changes in morphology or phenotype from these types of studies. One of the key findings that we found is that if you look at the dosing paradigms, you can tease out 2 different types of toxicity. One thing that the clinicians have found and identified for quite a period now is that the best way to dose a chemotherapeutic agent with a high dose initially, or to give it in smaller doses over a longer period of time. And what's been seen is that the high dose has been preferable because you get better patient recovery over time from the toxicity. If you give it over a longer period of time, there tends to be a prolonged cytopenia that occurs and a bone marrow toxicity. One of the hints for the mechanism that we see in this slide is that we start to see vascular toxicity occurring if we give multiple doses. So if you look across from the 1 dose, from 2 micromolar all the way over to the higher dose, you can see that the orange, which is the vascular cells, the CD31 positive cells, are fairly well maintained, even though you start to see a drop in both the erythroid cells and the neutrophils and the myeloid cells. However, on multiple dosing, you start to see the orange disappear. And so what's happening is that the vascular cells are now being damaged, in addition to the bone marrow, hematopoietic cells, and the myeloid cells. Coming back into the clinical response, what this shows is that what is probably happening with multiple doses is that you're not only damaging the myeloid cells, the bone marrow cells, you're also damaging the vasculature. And so that takes a much longer time for that hematopoietic vascular niche to reform. Whereas in a single bolus dose, you have a much faster recovery time because you're not seeing as much vascular damage. We've done a 27 compound screen across multiple chemotherapeutic compounds. This is a blinded study. And we did it in a manner of just looking at 3 different concentrations, a high, medium, and low. Some of the compounds, most of the compounds fell within the range that we tested. However, since we did it in a blinded fashion and we're just using standard doses, some of the compounds were either way out of the range, so we didn't see much of a response. So dose response was much higher concentrations, or we were well above what their IC50s were. And so we started to see toxicity just from the lowest dose that we saw. But of those that we were in the range, we predicted very well the toxicity. So an example is methotrexate. It's a standard of care chemotherapeutic agent. And so in this case, we looked at what was coming out of the bone marrow in addition to the damage that was seen within the bone marrow itself. And if you look at using flow cytometry, the numbers of granulocytes that come out, we see a significant reduction between 0.1 to 10 micromolar. And if we correlate that, we get an IC50 of about 1 micromolar or so. And with the clinical dosing, that typically is around 1.31 micromolar. And you start to see about 50% tox or cytopenia at that point, which is where most physicians start to maybe pull back on some of the therapy. So basically, we correlate very well with the clinical response. And it also highlights that we can utilize these systems now as if we're looking at potential cell markers in the blood, since we're mimicking what would be in the vasculature, different cell types that could potentially be used as biomarkers for toxicity or reduction, as well as, potential secretomics or other soluble biomarkers that might be coming out of these systems. The beauty of having, large amounts of this data is that we can now utilize this to feed it into machine learning as well as AI computational models. So as a first pass, we took the bone marrow data that we received for this toxicity of these 27 compounds and trained a convoluted neural network to predict what would come out, the flow cytometry data, and visualize a landscape of where we could see potential toxicities. And upon doing this, we can see this kind of 2D projection of a 3D unsupervised embedding using that neural net model. And we can see the clustering of green, which are the low toxicity compounds, and then starting to move into the red, which are the higher toxicity compounds. And what's interesting is that you now see at the bottom there kind of this fishtail where it branch pointed to 2 different toxicities. And what that indicates is that one path is seeing both the myopenia as well as the vascular toxicity. And then the other path is just seeing the myelopenia alone. So we're able to distinguish using these neural net models and these machine learning models, the different types of, and predict the different types of toxicities that we would see. And so the idea is that, a new compound coming in, we could look at the visualization of that phenotype as well as what's coming out in the flow cytometry and predict where it would fit into this toxicity matrix. Now, again, the generation of thousands and thousands of data points allows you to do very sophisticated analysis, and also allows you to make sure that you're utilizing things like machine learning and AI to actually do the analysis. Because really, when you're looking at thousands or 10,000s of different tissues, trying to do that manually, annotate and analyze all that is really beyond what most people will be able to do. So for like organ scale 3D phenomics, here's an example of, where we can optimize and then skeletonize the vasculature and then look at multiple different parameters. We can look at average vessel diameter, branching points, how many, the density it occurs. In addition, we can look at feature qualifications that we can use for rich data for AI inference models. And as I mentioned earlier, we can sample the effluent from every one of these individual tissues. And we can look at proteomics analysis, as well as any kind of secretomic analysis, transcriptomics, any kind of genomics analysis. So it really lends itself to really high throughput, high density, high content information that can be fed into the AI models from these human realistic tissues. As I mentioned earlier, one of the first indications that we use artificial intelligence and machine learning for is the automated feature quantification. As I mentioned, it's very difficult to do this manually, so we really leverage artificial intelligence and the ability to look at things like masking for vasculature, or looking at DAPI to look at cell nucleation in cells, as well as immunohistochemistry associated with those cell boundaries to identify different cell types and whether those are up or down regulated. One of the issues, looking at some of these models and the perturbations that you can do is the causal versus associative analysis. How can we get to the root cause of what is causing the toxicity? Or in some cases, if you want to utilize these models for efficacy also. Here's an example that we recently did of knockouts in a basic cell culture, and it really produces shallow data. So we use CRISPR to knock out engiogenic modulators in these 2D cultures of endothelial cells. And if you look across every one of these, I think you'd be hard pressed to see what any differences at all that there might be. Although we know that each one of these is a key critical factor in blood vessel formation and maintenance. Now, if you put these into our 3D model of the vasculature, you can now readily see, what the phenotypes are, the differences that each one of these endothelial specific receptors does. So with VEGF, for example, the knockout causes these kind of kidney-shaped vessel formations. They don't actually form vessels that connect with each other. Vehedron is known to be really seminal for keeping endothelial cells together. And so basically we see no formation at all. We see just cells, the endothelial cells kind of floating around in the matrix. So really see, what the phenotype can be. And with some of the other receptors, such as Tie1 and Ang1 and 2, we see different vessel morphologies. You see much thinner, much sparser vessels with some of the phenotypes. And so you can really start to tease out, what is the causal mechanism of each one of these different genes. And you can imagine that now we can extend this from this example to, multiple genes being now knocked out, or multiple genes being activated using CRISPR in different cell types, and seeing what the phenotypes are in our 3D human models. I throw this slide in just to show some of the other utilities. So we worked with our sister laboratory at the University of Pennsylvania, and we were looking at biomarker discovery for CAR T-cell therapy, and basically utilized metabolics to identify perfusates that might be circulating biomarkers that correlate strongly with a therapeutic effect for different compounds. And so you can readily utilize this system to look at blood-based clinical biomarker discovery for different cell therapies. So in summary, I hope what I've showed you is to close that translational gap. We really need scalable human biology. And the key here is enabling, very high translatable tissue relevant models, which have better human physiology than either low content assays or even organoids. And that allows us, with the throughput, to do multiple perturbations and optimizations to see which tissue types are generating specific biomarkers, or to verify the identity and or identify new biomarkers used in these systems. And as I pointed out, the biomarkers can be anchored in mechanism, not association. We can, make functional perfusible vessels, bone marrows, other tissues such as liver, airway, and lymph node, and identify clinically relevant readouts and space-specific gene perturbations to verify, whether the biomarker is true or not. And the idea is to, and I should have mentioned this earlier, is to find a biomarker that can be translated from preclinical studies in vitro all the way through to the clinic, often, and can potentially even be used in animal studies. Often that's very hard to find. They're kind of the gold standard. And so we're stuck with using either surrogate biomarkers, either in patients or not able to translate those from preclinical to predict if those same biomarkers would be useful in the clinic. We can use longitudinal blood-like sampling for translational biomarkers. We can do repeated puberty rate collections and support circulating biomarker discovery. And as I mentioned earlier, we can use transcriptomics, metabolomics, proteomics, looking at cytokines, and link those to a therapeutic response or a toxicity dynamically, and really to identify clinically actionable endpoints. In addition, we have AI-ready phenomics, as well as other, more deeply high-content versions with transcriptomics as well as proteomics to feed multimodal embedded NMI and interference models. We've given an example where we can utilize this to predict chemotoxicity. As you can imagine, you can utilize the same types of techniques to predict clinical outcome for efficacy, as well as clinical outcomes for safety for other different tissues. And what this allows for potentially is biomarker panels for patient stratification, especially for safety de-risking, and hopefully ultimately for clinical trial design. Just like to acknowledge my fellow members of the lab, Andrei Georgescu and Dan Ha, who founded the lab, and John, who is the director of biology and did a lot of the work, and the entire Vivadyne team. As you can imagine, developing these types of systems, whether they the biology or the hardware or the software, requires a multifaceted, multifactorial team of a lot of different expertise. And so, there's too many to mention in this slide, but we have an extraordinary team, and I'd really like to thank the Vivadyne team. And with that, I'd like to take any questions.
Thank you very much, Tony, for that wonderful presentation. We'll now move on to the Q&A session. It's a reminder to the audience to submit your questions using the Q&A system in the live stream player. We already have some questions, so we can jump into those. First question is, have you observed or tried to introduce osteoclasts, osteoblasts, or osteocytes, or bone marrow adipocytes into the bone marrow model? And if so, what challenges have you faced to make a fully mature bone marrow model?
So can you hear me okay?
Yes.
Yeah, so to date, we have not tried to develop an osteogenic bone model. That's kind of the next step that we'd like to initiate and develop. So the short answer is no, but it's something that we're certainly thinking about to do in the future. We have not added adipocytes to this current version either. It's an interesting idea, and potentially a future area that we might move to. But no, we have not done that yet.
Thank you. And a follow-up question from the same audience member: have you used a multi-organ system?
We have. One of the examples, and this was in a paper published with our University of Pennsylvania lab, is where we've interconnected the large airway model along with the bone marrow model. And in that instance, what we've done is infected the airway model with bacteria which then released, cytokines. And then the bone marrow basically released neutrophils and white blood cells to travel to the site, and those were able to translocate across the epithelial barrier and attack the bacteria on the apical surface of the airway epithelia. An interesting thing there is, one of the things that we noted was this kind of swarming and netting that was being shot out from the neutrophils onto the bacteria, and so we consulted with some of the clinicians, and we initially thought this is an artifact, and I guess a few weeks prior to us seeing this, there was a paper that was published, and they actually were showing that these neutrophils were kind of shooting out these nets and arrays to trap the bacteria. So it's kind of interesting that we saw something that was unique to physiology in our system also.
Thank you. And next question is, can this be adapted to a glioblastoma 3D culture?
Yeah, that's an interesting question. So we have done tumor models where we've embedded in the ECM. We have not, done neural tissue yet because we haven't expanded into utilizing to a great degree iPSC cells. So it wouldn't be say a neuroblastoma in the context of say a neural cortex or a neural, tissue. What we can easily do at this point, and we haven't done it yet, is potentially utilize it in our blood-brain barrier model, where we have the brain microvasculature, the parasites, astrocytes, microglia, all associated appropriately in an extracellular matrix, and then place that glioblastoma in that, and then look to see if different therapies can reduce the size or reduce the growth of that neuroblastoma. So that would be a kind of a very easy next step for us to accomplish. I think putting that into a context of just neural tissue is certainly doable, but would take a little bit of development.
Thank you. And the next question is, what's the advantage of your platform over organ or tumor on a chip?
There's two that I can think of right offhand. One is that we are truly using vasculature to perfuse the tissue as well as the tumoroids. Often, kind of the organ on chip models are kind of plastic tubes that are lined with endothelium, and so that's kind of mimicking your vessel. And then the epithelial is on the other side of a permeable barrier. So it's typically limited to a barrier model and not a true 3D tissue. The second one, and I think it might even be the most important, is the scale. Most of the systems that are out there, either in academics or commercially available right now, are relatively low throughput. Still lots of manual labor, even those that are kind of semi-automated. The advantage that we have is that we can look at tens of thousands of these chips independently and do this on a fully automated system that requires very minimal human intervention. So the reproducibility is extremely high compared to some of the manual organ on chips, where you can get user to user variability very easily. And the scale allows you to do things to optimize tissues as well as interrogate models, interrogate targets, which beyond just the whiteboard exercise. You can often come up with tons of ideas for experimental protocols or evaluations, and then you're limited by the exact experiment that you want to do. The scale that we have really opens up the possibilities. You can just do the experiment and let the data drive where you want to go and what the decisions are.
Thank you. And can you provide some additional insight, please, about the possibility of adjusting oxygen levels in the system?
Yeah, there's ways that we have, outside the hive, incubated cells at various oxygen levels. The hive itself is fully enclosed and it's 5% CO2 and normal oxygen levels. We could potentially change that and have certain hives with lower oxygen levels, but that be an extreme case. I think the better option there is that we could potentially utilize materials that aren't as oxygen permeable, and then utilize kind of a chip design and microfluidics to control the oxygen tension levels within the individual chips and the individual tissues.
Thank you. And the next question is, why do vascularized human 3D tissues provide more clinically relevant biomarker readouts?
I guess the simplest answer is that they represent more faithfully an anatomical structure, the cell-cell interactions, as well as the physiological functions of human tissue versus 2D systems or potentially even animal models, because they are human-based. So I think the more translational, the more that they mimic the true human physiology, the better biomarkers that you can generate out of those. And because of the close compactness of the cells, I mean, we've got 200,000 or 300,000 cells. They're very closely associated with each other, and they're signaling between each other in an appropriate manner and giving each other the appropriate cell signaling. I think you're more faithfully recreating the physiology and the human physiology there that you can then interrogate to see which might generate better biomarkers for either disease, progression to disease, and or reversal, trying to modulate those and bring disease tissue back to healthy tissue.
Thank you. And can you provide some insight about the platform's ability to incorporate aerosol exposure at the ALI, or is it currently limited to liquid phase dosing?
Yeah, we have not incorporated nebulization or aerosol delivery at this point. Most of our exposures are through the vasculature mimicking IV. There are ways that we could potentially do that. We've been asked by different partners whether this might be feasible, and I think there are ways to provide lids that cover the epithelial tissue and the airway tissue and allow for kind of aerosol delivery and nebulized delivery directly onto the epithelial apical surface. Again, I think that would take some slight bit of engineering, but I think it's certainly possible.
Thank you. And there's a question from an audience member about whether academic researchers could send over compounds to be tested on the automated system similar to a CRO model.
Yeah, that's really not our business model. We are, really, as a company, looking to make partnerships and collaborations with biotechs and large pharma. We are not a CRO, so just to make that clear. We're not on a fee-for-service basis.
Okay, thank you. The next question is, how does high-throughput automation improve reproducibility and biomarker credibility?
I think for the reproducibility, it's not so much the high throughput, it's the automation. Often, we've heard stories and actually have had, instances even within our own labs where you can have two people, at the same lab bench doing the same, type of model, and just the ways they may pipette or the angle of pipette or the way they do the procedure, you can get very different outcomes, on some very subtle changes. The robot does it exactly the same every time. And so, the robustness and the reproducibility and, the standard errors and the standard deviations, really dramatically get much smaller there. So the reproducibility is greatly enhanced just by the automation itself and the robotics. The throughput allows you more to, test multiple parameters and put you in spaces, especially, if you're looking at the preclinical development pipeline, much earlier in the process also. So you can test, thousands of compounds in preclinical screens versus, being limited to, say, lead optimization, where you're maybe looking at, 3 or 4 compounds, but you want to choose the best one going forward. But the automation, I think, and the robotics is what really reduces the reproducibility or increases the reproducibility and the fidelity. And that allows you to maybe tease out, subtle changes and subtle biomarkers that, might be lost in the signal to noise otherwise.
Thank you. And we have time for one last question. How do you define and defend a biomarker's context of use early on before it gets embedded in pipeline decisions?
That's a great question. I think that you would almost have to go through the same procedure, say, like the drug development tools, and qualification that the FDA uses for, human clinical biomarkers. So you'd have to have good faith in, why preclinically, it's associated with the either disease process or, a toxicity. Move that forward and then, test that in patients and see if that does translate. So, a novel biomarker is, quite a process to validate or, in the FDA's terms, qualify.
Thank you very much, Tony. We really appreciate you joining us today. We will be moving on to our next session shortly with Arash Zareen Afsar.
Thank you.
I'm pleased to welcome Arash Zareen Afsar, who is trained as a physical biochemist with interest developing various spectroscopic and spectrometric methods to understand the dynamics of biological systems, in particular, transition to the disease state. He's an associate professor with the Department of Surgery and Graduate Department of Medical Biophysics at University of Toronto's Faculty of Medicine. He's a principal investigator and leads a group of researchers focused on development of rapid diagnostic methods using mass spectrometry. His lab's research over the past 5 years has further matured picosecond infrared laser mass spectrometry as a rapid pathology determination tool, where 10-second laser sampling of tissue molecular content has allowed differentiation of cancer types and subtypes faster than currently possible in the standard of care. Welcome, Arash. The floor is yours.
Good afternoon, everyone, and thank you for attending my webinar, and I appreciate the opportunity by organizers to walk you through some of the developments from my lab on the utility of lipidome analysis for rapid cancer diagnosis. I have some disclosures related to inventorship and creation of startup in this space. Proteins have been our friends and diagnostic partner for decades. Disease states such as cancer often results in differential expression of certain proteins that are used as biomarkers. And the same argument is applicable to the genome. And unfortunately, metabolome has been a little bit understudied when it comes down to its utility for cancer diagnosis, aside from a number of oncometabolites here and there. And I think part of the reason is that the chemical diversity of metabolites has really scared people away, and it's really hard for you to have, as I said, there are cases where there's a single oncometabolite that's highly diagnostic, but I think with a little bit of tweaking the notion of biomarker to a metabolite array, or a lipid array, a list of lipids and metabolites that are characteristic in alterations of abundance for cancer, they will have utility. And especially with the method such as mass spectrometry, you could much more quickly access metabolic changes. So you reduce your time to diagnosis drastically if you can you can revisit the notion of metabolites, and I use this in a plural sense, as biomarkers for disease states. In particular, lipid metabolism is tightly coupled to cancer formation. Cancer cells are hungry for energy, and they upregulate fatty acid synthesis and uptake. There's also phospholipid dysregulation. Sometimes phospholipids are broken down to fatty acids that are then used in beta oxidation to create more energy. And consistent with this general trend, a number of enzymes that are involved in lipid biosynthesis pathway are now considered potential drug targets for certain cancers. One of the methods that allows quick access to tissue lipidome is ambient mass spectrometry. And the idea here is that if you have a desorption or ionization or a desorption source or extraction source that allows you to very quickly lift your lipidome from the solid tissue directly without a lot of sample prep to the gas phase, whether you post-ionize it or not, different story, you could create these lipid profiles that are MS1, highly superficial, equivalent to direct infusion. So you have to be very careful with how you interpret this. You can't really be confident because of lack of chromatography separation that you're dealing with single molecules that all of your MOVCs, of course, depends on your resolving power, the mass analyzer you use, and so on and so forth. But one of the leaders in this field is Livia Eberlin, who has created the mass spectrometry pen, which uses a drop of water or solvent to create this extraction and accesses tissue MS1 very quickly. And she published this really nice paper where she walks us through different desorption methods. And I've highlighted some here for you that you will see in the literature. iKnife, intelligent knife, is one of them. It's an electrocautery blade. It was developed by Zoltan Takacs and now being worked on by Waters. So it's an electrocautery blade that's got its smoke evacuation tube attached to an interface for rapid evaporative ionization and online mass spectrometry. You also have infrared lasers, such as nanosecond source by Isabelle Fournier and Michel Salzé that do the same purpose. The advantage really of these ambient methods is the speed of analysis. My lab uses a variant of these methods called picosecond infrared laser mass spectrometry, and the idea here is that we deposit significant amount of energy into the hydroxyl stretch vibrational mode of water, and because of the intrinsic coupling between the OH stretch and ablation liberation modes, most of the deposited energy vaporizes the water molecules, and we know that biological tissues are rich in water, so this explosion basically lifts whatever else that tissue that was dissolved in tissue water, if you will, for lack of a better word, such as lipid, proteins, chunks, and all these things. So there's a lot of parasitic processes going on at the same time, but you also produce significant amount of gas phase species, and you can just see that this plume expands very quickly in the air. You could just see the shock wave by dark field imaging, and consequently you don't need a lot of tissue material. And in terms of ionization, if you just keep your plume content over the resolvation potential, you can just thermally maintain them in the gas phase species, and you can create a mass spectra. And we decided deliberately to stay away from ionizing or using post-ionization to further enhance our abundances, and we take this trade-off. We trade intensity or abundance with diversity, and we read our lipid profiles in a negative ion mode for all the things in the tissue that were pre-charged to begin with. And the idea here is that we want to remain as faithful to tissue biology as possible. So the moment you invoke post-ionization, yes, if you want to do targeted analysis, it's great. You want large, fat, big peaks, but competition will take in that could result in some simplification of your spectra. Things that ionize really well will always want to be ionized, so you're losing some of your depth. The workflow of analysis is quite simple, as I said, and justified why we need to look at our spectra over the range of fatty acid and phospholipids. So we record our spectra over 10 seconds, and we generally bin them to 0.1 Daltons, and you could do a very quick multivariate analysis. So if you have a library of what is in differential diagnosis in the multivariate space, you could, it's not computationally expensive at all to do, let's say, principal component analysis that takes milliseconds on your device, and by way of positioning of your Cori spectra in the multivariate space, you can reach a diagnosis. And where we're going with this is we've taken our inspiration from genomic assays, and there are transcriptomic based methods using nanostring sequencing, for example, wherein a large number of transcripts are empirically determined to be upregulated, downregulated in certain cancer classes, and we read the abundance of these transcripts using a quantitative a sequencing method, and we want to do the same thing with lipids, but the advantage really is that once you have this lipid array, you could access it much more quickly than your genomic assay. So you go down to a matter of few seconds from something that takes hours to collect. Eventually, we want to create a tool that we could put in the hand of our surgeons, and our application has been mostly around neurosurgery, wherein they could do the sampling themselves. Of course, you need to have a pathologist for sign-off and validation understanding. So you lift your lipid signatures. There are some water molecules. We don't really show them they're present, but they're kept above the desolvation potential. You do a multivariate composition. So if you have a library of what these MS1 patterns will look like, then you would be able to arrive at diagnosis very, very quickly. One of the first things we did was we assessed the the potential of the system for in vivo measurements. We took a bunch of mice, put them to sleep, opened up their skull, and did craniotomy, and we recorded the live the mass spectra of brain from live mice under anesthesia, and the signatures in a multivariate. This is a PCA-LDA scores plot corresponded to what we got from frozen tissue. We had the opportunity to sacrifice these mice and subject their slides to diagnosis for degree of damage. So obviously the volume that you've removed with the laser is gone, but you don't really have a lot of damage leaking, so you can read histology 25 micron below your crater. With respect to DNA damage with tunnel, apoptosis, cell death with caspase-3, and neuronal viability with new and staining, we did not really see drastic differences between laser and a scalpel. Of course, you create quite a bit of insult with the laser with respect to DNA damage that recovers very quickly. But the bottom line of this experiment is that we are able to use this source compared to a scalpel, which is widely used in neurosurgical application without concern for tissue damage. We also dabbled with how we're going to display the pathology results. So this is a proof of principle experiment we did. We coupled our laser to neuronal navigation system. These are LED balls that are tracked. So it's like a GPS for your tool and coordinates. So you could display your augmented reality view. And here we've created this artificial boundary between healthy and disease tissue, and we have a molecular model, a mass spectrometry-based lipid array model for what healthy and diseased tissue looks like. And every 5 seconds, we are parsing and asking the question whether this average of 5-second mass spectral worth of data is disease or healthy. And then once the classifier is made, then we display that in a form of a blue or red indication to the surgeon. But everything that I'm going to talk to you about in the next phase of the talk has been completed with ex vivo tissue. So the cancer is excised from the patient and is brought to the mass spectrometer for analysis. And I'm going to walk you through 3 clinical cases. Case 1 was around molecular classification of pediatric brain tumors. And this was a work with Jim Rothka, Mike Taylor, David Munoz, our neuropathologist, and Howard Ginsberg, our biomedical engineering and neurosurgeon. And the reason we wanted to do this is that there are several molecular subtypes of these cancers, and they all have very different prognoses. So if you look at the major classes, ependymoma, pilocytic astrocytoma, and medulloblastoma, the gross total resection, how aggressively you need to resect, is highly subtype dependent. And if you look at the medulloblastoma, for example, there are 4 major molecular subtypes that are morphometrically identical. So neurosurgery is an interesting case because you don't generally have preoperative biopsies when you go to the surgery. So when the patient is exposed, diagnosis needs to happen around in about 20-30 minutes or so, and you can't really do a molecular classification. So morphometrically, using a hematoxylin and using a staining, which is common for intraoperative consults, you cannot resolve these molecular subtypes. But if you look at their respective analysis of the prognosis, group 3, very, very poor prognosis, gross total resection, does not really enhance the survival. On the other hand, W and D, very good prognosis to adjuvant therapy. And also gross total resection is not beneficial. But if you have a group 4 medulloblastoma, maybe there is an argument to be had for gross total resection. So how does the surgeon know what molecular subgroup they're dealing with? And again, I don't want to bore you with going through the classes of ependymoma, because they cannot distinguish these molecular subtypes on time scales of hematoxylin and using the stain. So we got our hand on about 120 medulloblastoma. I think overall a patient used for this cohort was about 200, And we recorded our 10-second mass spectra, and sure enough, we could actually see some differences. And I know you can't quite see it from these representative spectra, but once I show you the model in the next slide, would be a little bit more clear. We also wanted to make sure that the molecular models that we use are containing sufficient diversity. And one of the things that we look at is we look at the performance of our molecular models as a function of independent sample numbers that are used in them, and we sort of look for the plateau. We kind of reached a plateau at about 100 sample use, and our study is going up actually over that. So we think that we are statistically powered to make significant judgments. So this is a multivariate principal component analysis, linear discriminant analysis of our data set across the classes of pediatric brain tumors that I have shown here. So we have pretty good separation of the classes. Each of these balls is a 10-second data point. With respect to the medulloblastoma cohort, we also have a differentiation between our 4 subgroups that are currently impossible. So we propose this two-way classification wherein we first subject our unknown to this model, and if it comes back medulloblastoma, then we're going to use different model. And these models use different ranges of lipids, so you can kind of mix and match your classifier. And sensitivity specificity was validated using a blind test and was proven to be greater than 94%, which is great. The other interesting thing in this study was that regardless of where we sampled on the tumor core, and I admit these are tumor cores we're looking at, so the issue of margins and healthy tissue contamination completely secondary, the concordant rate was about 91%, which is pretty good. So by going to use of lipids, we're desensitizing ourselves to some of the clonal evolution that you could see in, let's say, metallomic profiling here, which is a good thing, because eventually you don't want to have a tool that you get different results from based on where on the surface of the tumor you did the sampling. You could also try to optimize your classification by making the model more stringent. So here we drew these are probability hyperplanes that you can draw to sort of define what belongs to one class or not. And if you're making your model stringent, you could just see that your number of misclassification goes down, but the number of unclassifiable, the model tends to say, I don't know what it is, a lot more. Of course, you don't want this number to be too high, because a tool that can't tell you, half the time what it's dealing with also does not have a favorable duty cycle. But clinical burden of misclassification is much higher than clinical burden of unclassifiable data. I mean, unclassifiable data is the status quo. Right now, there's no way you can molecularly classify these things on intraoperative time scale. So, I mean, you can live with this, but you don't want to have significant number of misclassification, because that's where there's a burden of incorrect treatment or suboptimal treatment for the patient. So remember, I mentioned to you that it's very challenging for us to refer to our MS1 profile for direct tissue extraction in context of biomarkers. So we do orthogonal laser capture microdissection, HPLC, UPLC, MSMS analysis, and we've convinced ourselves that our laser plume, which is captured on a filtered paper, is almost equivalent to a lipid extract that is captured directly from the tissue. So we can trap the laser plume and do a biomarker discovery and sort of link the data sets this way to identify our arrays. And here's what we've done. Again, I don't want you to go through all of these numbers. Suffice it to say that we created this metabolite array, which we identified our identities using UPLC of the laser plume, and we went back and recalculated our sensitivity specificities only using this lipid array. And you can just see that there are cases where we do much better than the others in terms of sensitivity and specificity, but overall the results were actually quite promising. The second clinical case that I want to walk you through revolves around the utility of this method for discrimination of common spinal tumor types. So spinal tumor types are diagnosed using this kind of decision tree based on whether they occur extra to the dura or intra to the dura, and whether they are actually intramedullary or extramedullary. And in terms of the common types, you're dealing with metastatic cancers, meningioma, and schwannoma. So why do we care about this clinical case? We care about this clinical case because schwannoma tumors are derived from Schwann cells. So in a way, the nerve becomes a capsule to the tumor. So for you to resect the tumor, you really have to sacrifice the nerve. Meningiomas, on the other hand, you can take your time and dissect around the nerve. There's really no need for you to do a quick excision. They are non-malignant, but they need to recur. So the complete resection with dural excision is required. So if you have a competent neuropathologist on call, you don't have a problem with telling these classes apart. But often enough, you get a lesional tissue diagnosis, and as a neurosurgeon, you're stuck. So you tend to do a very conservative surgery and call the patient back in once the permanent pathology results are made available, which could be weeks after your initial surgery. So the value added proposition for our research here was, could we very quickly tell the surgeon they're dealing with metastatic meningioma or schwannoma using our molecular profiling? So we got our hands on, and this is the work that was published in Neuro-Oncology a few months ago, we got our hands on meningioma, metastatic schwannomas, recorded our 10-second spectra. Again, these are the sample numbers. And we had very good distinction based on lipid profile. So these these are score plots, again, PCA, LDA, principal component analysis, linear discriminant analysis. This uses the entire worth of your mass charge ratio. So we haven't identified our lipids yet. We don't have our array yet. But we wanted to convince ourselves that this separation is real. So you mix your classes, create a permutation, your cross-validation from fivefold, your accuracy from fivefold cross-validation goes down to 41% from 98. So this separation is somewhat real. And again, the same with the learning curve. We seem to have reached some sort of a saturation at about 80% of our data use. And this is encouraging. So we went ahead, again, the same story, UPLC, MSMS, laser plume, and we identified, if I'm not mistaken, around 41 lipids. And we were able to go back and validate the prediction of this model using these extra blind samples that we accessed. So one of the things that we did here was we went back and added other classes of spinal lesions that are informal diagnosis. So remember the previous model, these biomarkers are coming from this list of a 3-class meningioma, metastatic schwannoma, which is the prominent ones. But if you keep adding other classes in differential diagnosis, your model performance stays the same. These are so different than the other 3 classes that their presence makes no difference. If you look at the confusion matrices, for example, you don't really see any misclassification between these new classes, which are also informal diagnosis with meningioma, schwannoma, and metastatic cancers at all. So as a next step, we actually went back and tried to reanalyze our data using the lipid array that I just talked to you about on the previous slide. So here we're showing the result of our 5-fold cross-validation, only looking at the lipid array. So remember, if you look at the entire mass charge ratio, you had a 98% cross-validation. And by using the lipid array, this actually goes down to about 91%. So they're clearly, we're not capturing some of the information there because the lipid array was designed using only the major classes. So there's an opportunity for improvement, but it's actually not so bad. The second clinical case that I'd like to walk you, the third case that I'd like to walk you through about is the utility of lipid profiling for guiding the extent of resection in neurosurgery. So if you are a neurosurgeon, you spend a lot of your time trying to determine whether you're dealing with a glioma, lymphoma, or metastatic cancers. And lymphomas are quite rare, but they're really the odd one out because they don't really need any resection. They respond really very quickly to adjuvant therapy. So metastatic and gliomas have different extent of resection to optimize outcomes. And there's also some molecular classification for glioma you need to be aware of based on the isocitrate dehydrogenase status. So can we use our technology to help guide extent of resection? And we did the same thing, got our hands on a number of glioma, lymphoma, metastatic cancers, sample numbers that I've shown here, do a usual thing, make a model, make a permutation of it, look at the learning curves, make sure we have enough samples and everything checks out. We went ahead and looked with UPLC MSMS, it created these metabolite array, lipid array, lots of PEs and PCs and ceramides involved, and went back and did a cross-validation fivefold using these 32 lipids. So if you use the entire worth of mass charge ratio, you have a 93% accuracy, go down to 87. So this is all quite exciting. And most important thing for me was that we actually were able to demonstrate looking at 2-hydroxyglutarate as an oncometabolite that's associated with IDH1 status, using a cohort of around 100 tumor types per class, we actually had an accuracy of almost 87%. So we actually see 2-hydroxyglutarate with a loss of water in our case. And it's kind of interesting because at least for some low-grade gliomas, the survival of IDH1 mutations depends on really the aggressive resection. So this really justifies the extent of resection in a way that takes into consideration our rapid utility of IDH1 mutation. And we also went ahead and created a number of orthotopic tumors in mice, and we sampled them in situ post-sacrifice, and we showed that, yeah, we could tell glioblastoma from breast cancer and lymphoma. Again, breast cancer and glioblastoma are representatives of our metastatic carcinoma and glioma cohort. So there is potential for this method for future use. But how are we going to use this method when we deal with very, very large data sets? So right now we're sitting at about over 1,000 spectra from over 10,000 spectra from over 1,000 patients across 24 different classes of cancer. I'm showing you a UMAP distribution of our plots. Again, each of these dots is a 10-second parallel MS spectrum. So we're lucky because pathologists never really look at everything in one go. I mean, they go through these so-called decision trees. So if you look at the WHO, World Health Organization Handbook of Clinical Diagnosis, you see a lot of these decision trees that are put together. In this particular case, I'm just showing you a small tree for diffuse infiltrating gliomas that are first classified based on their IDH1 status. So you can envision a mass spec-based assay. The first thing you look at is your presence of 2-hydroxyglutarate for IDH1. Then you know where you go left or right in this tree, and then you can invoke different models for each node. And this simplifies really your diagnosis. So for our data, we put this large tree together. I don't want you to, learn from it. Suffice it to say that there are nodes and classes and subclasses, and each nodes allow you to incorporate multimodal diagnosis. For example, for most of the brain tumors, you do have an idea whether they're intramedullary or extramedullary from some MRI images preoperatively, so it can simplify your diagnosis. So what is the advantage of this hierarchical tree base? I mean, first and foremost, it does simplify the number of classes that you must simultaneously distinguish. You can use different models across each node. So remember, if the first node is IDH1 status, you don't even need a multivariate analysis. You just do a univariate, set in your oncometabolite, and you're done. You can also adjust the depth of diagnosis according to a desired performance criteria, and you can also consider the cost function for misdiagnosis. So what do I mean by these two last concepts? I mean, look at this tree. This is the tree that we discussed for surgical planning of tumors, right? Glioma, lymphoma, metastatic. You can break your metastatics into organ of origin. Of course, the resection extent for these is all the same. It's just, the adjuvant planning is different. Or gliomas, need to be resected differentially based on their IDH1 status. So, what do we mean by adjusting the depth of diagnosis? Imagine this tree. You can look at it, you're like, well, I'm just going to stay to node A, the top, and say this is metastatic carcinoma. I don't think I like that I have only 65% confidence to say it's coming from lung. Yeah, it's probably coming from lung, but I'm just choosing to say, hey, I only can tell you confidently that's metastatic, and I'm not going to go down this fine branch. You could also apply this cost function to address misdiagnosis, and this is the benefit of the big picture, holistic, or adaptive decision making that clinicians use. And sometimes as mass spectrometrists, we get bogged down by numbers, and I'm not saying that the statistics are not important, but again, assume these numbers there's no error bars and they're all significant. So if you look at this tree for node A, everybody's going to go based on lymphoma, right? But the clinician could say, well, I am just going to go based on metastatic because the cost, the burden of misdiagnosis, metastatic as lymphoma, is higher than the other way around because lymphomas don't need to be resected. If you end up resecting a lymphoma from a patient, you've just done unnecessary work. But if you end up not resecting metastatic cancer, the patient's going to be in a lot of trouble. So this is the advantage of this hierarchical approach. And again, I don't want to bore you with a lot of details, but we have this workflow put together wherein we are breaking up our data into test training sets, generate node-specific data matrices and optimize a number of machine learning methods for each node. This is a project that by my PhD student, Dara Flamick, and our collaborator, Scott Hopkins, who's a mass spectrometrist data science guru. And I'm not going to show you a lot of data here sticking to the node A. So most of these machine learning methods sort of perform similarly well. This is our balanced accuracy estimates with standard deviations as the error for your fault validation. But they did so by looking at very different feature lists, and this is kind of important for us because features eventually are related to biomarkers, right? So feature extraction is important. Yes, all of these methods work, but they kind of see different things in your spectra, so you really need to understand what these features are and what they mean. And where we're going with this eventually is trying to create a graphics user interface for our pathologists to use based on this method. So a lot of it is hard-coded. Your lipid arrays in the background, you've optimized it. You have the tree in the background and a lot of other technical things. Your binning, your log mask correction for instrument drift, especially use the time of flight, goes in there. Spectral QC, quality control, is a big one. We're trying to mine our data sets right now from a computational and experimental point of view for what is a good quality mass spectrum. Is it, number of peaks you're looking for, presence of peaks in expected regions, let's say phospholipids, fatty acids, and or is it just noise? We're approaching this from empirical dehydrated tissue, which can happen, detuned laser, all these things. So those are experimental bad data. We're trying to mine to see what they mean, because what we want to do is we want to make sure we don't send in garbage spectra and get garbage classifications back. And we also engage our pathologists in user experience. How do we display the features that they use? Like, do we want to see decision tree? Do they even care to see the mass spectra? Probably not. But this is a collaboration we have with our cancer digital intelligence group. And we're also looking at unsupervised decision trees and word linkages. And this is the same data set that's now separated into this tree based on unsupervised analysis and distances between the data. And the idea here is that we're going to go from nodes that are very highly discriminatory from one another to make our strategic binary left or right decisions. And one of the things you could do with this is create this data dependent tree, and you could just see that how much improvement you get in your balanced accuracies if you compare the classes based on a 24-way one-shot classification to an optimized tree. Of course, there are cases where you're still suboptimal, and you can look at the confusion matrices. In this particular case, you're 30%, 6% for this node, and you can try to see what the culprit is. Maybe take it out and put it somewhere else in another tree where there's no chance of crosstalk, but or stick to the higher branch, which is quite reasonable. So bottom line, there's a lot that we need to do, and on this stage, I'd like to thank my staff and funding agency.
Thank you very much, Arash. What a wonderful way to round out this event. Unfortunately, that's all the time we have for today. However, any questions that we didn't have time to get to will be answered offline as soon as possible. All talks from today's event will be available on demand for the next 30 days. You can access them using the same link you used today. We'll also make sure any questions submitted on demand are passed along to the appropriate speakers. That just leaves me to thank everyone who attended, as well as our speakers and sponsors. We look forward to seeing you soon. Bye-bye.